So our next speaker is Bjorn Merker. Bjorn is an old friend of BTCB13. This is his third talk here. And so he's been a great supporter of what we're trying to do here, and also gives tutorials when he comes. So it's great to have him back. Bjorn told me just now that he was born in Bohemia before moving to Sweden, and but as a true Bohemian, I think, in the wide sense of the word, he also spent a large part of his youth traveling in places like India, where he had the insight that the thing he should really do with his life is try to understand the brain. And he's here after a long career, mostly in America, of doing neurophysiology to try and understand the brain experimentally. And then he retired from active experimental work and went to live almost as a recluse in Sweden and think about the brain, and began writing. And after a period of 10 years, he has emerged again, I guess rather like a butterfly out of its chrysalis, and he's sharing with us in his talks at BCBT some of the insights that I think he's developed over this very long career, combining experimental approach with what we agree could now be called neurophilosophy, which is how he sees himself. So I'm really looking forward to hear what Bjorn has to say about the midbrain, really now, the superior colliculus. Bjorn.
Thank you so much. Oh, I should put on this shirt.
Yeah.
Okay. It's green? Yes, now okay. Thank you very much, Tony, for that wonderful introduction. I didn't know I had such an exciting life, but it's great. So this is going to be a little bit touch and go. Yesterday morning, I woke up with a flaming throat infection, and it derailed my whole day. I cured myself yesterday, tried to cure myself by surviving on 5 apples and 2 bananas, plus more than a liter of Coca-Cola. I call it the elixir of life. And that moved the infection from my throat up to my nose. So now I have equipped myself with tissues. It also has affected my brain. It feels like my brain has gone through Leah Krubitzer's isotropic fractionator, or as if there is cotton wool stuffed into my head. And luckily, I had my talk written out mostly. And so I had the write-up of my talk printed out, and I will actually rely quite a bit on this, because if I start talking extemporaneously, I think it will be a mishmash. So to keep on track, and I think I will definitely keep on time, I've run this through once, and it should be about 55 minutes, and then we have time to questions. So this lecture will differ a bit from other presentations here, in that with one small exception, I will not be presenting any data of my own. I'm in retirement and I'm currently not engaged in empirical studies. Instead, I will review a functional issue, the role of the superior colliculus in the brain's operational division of labor, and some of the changes the interpretation of that role have undergone in the more than 150 years that this structure has been studied empirically. For those who know the colliculus largely from textbooks accounts of it as a visual reflex center for eye movements, there will be some news in my talk. But for those who have followed the work of Douglas Munoz, Richard Krauslis, Jeffrey Schall, Robert McPeak, and others on the monkey superior colliculus, my talk will bring no earth-shattering novelties, though even for them I hope to provide some useful context and perspectives. I did my thesis work under Jerry Schneider in the Department of Psychology and Brain Science at MIT in the late 1970s. The topic was the behavioral role of the colliculus, which I studied in hamsters using behavioral and lesion methods. Here is the title of my thesis, which I finished in 1980. A lot has happened in colliculology, we can call it, since 1980, and over the intervening 33 years, I've tried to keep abreast of developments out of the corner of my eye while doing other things. But recently, I found myself plunged headlong into the question of collicular function while working on the following recently published paper. In the course of trying to understand and to integrate a number of disparate strands of recent evidence regarding collicular function, I came to realize that after a century and a half of empirical studies devoted to that topic, the leading edge of operational constructs designed to capture its distinctive role in the brain is finally starting to match the incredibly sophisticated anatomical circuitry that is housed in the two little bumps that crown the rostral end of the midbrain. We will see them in a moment. It looks to me, in other words, as if with the current suggestions regarding the functional role of the colliculus, the long historical trajectory of its functional interpretation is reaching some kind of asymptote. And I thought I would tell you about why I think that is the case. So let's start with the two little bumps on the top of the midbrain. Here is a human brain stem seen sideways in a slightly oblique view. So here you have the pons and the medulla and the spinal cord. Here is the cut brachium of the cerebellum. So the cerebellum would be sitting like a huge ball here. And of course, the cerebral cortex would be a huge mushroom-like thing surrounding this whole thing. Here is the thalamus. And here is the midbrain. Inferior colliculus. You can see the pointers, yeah. Inferior colliculus down here. Superior colliculus, just a little bump. This thing is not the second colliculus. This is the pineal gland between the two colliculi on the left and the right side. So that looks like a very inconspicuous thing, these two little bumps. Now, these two little bumps, they may not look like much, but the grandmaster of the comparative neurology of pre-vertebrate ancestry, Thurston Lacalle, those who don't know his work, I highly recommend it. This man, who spent his life studying pre-vertebrate and the transition from pre-vertebrates to vertebrates, he called those two little bumps, I quote him, "The quintessential identifying feature in vertebrates is the dorsal optic tectum." The quintessential identifying feature in vertebrates is the dorsal optic tectum. He did not make that claim lightly because he thought he had identified the homologue of the vertebrate tectum in the pre-vertebrate amphioxus, the lancelet. And he published that claim only to have to eat his words on further study, a very embarrassing thing for a scientist. So only vertebrates have a tectum, called a superior colliculus in mammals, or anything remotely resembling a tectum, and all vertebrates feature it in a remarkably conserved fashion as far as its structure and connectivity is concerned. The same cannot be said, for example, about the glomerular organization of the vertebrate olfactory bulb, which shows some striking parallelism to that of the insect antennal lobe, which we heard both yesterday from Mombarts and from Bill Hanson today. It even seems conceivable to me that in light of the functional arguments I will review, the presence of this little structure in the roof of the midbrain of all vertebrates, and only vertebrates, may have something to do with the rather remarkable evolutionary career of vertebrates by giving them a specifically vertebrate solution to a functional problem other phyla must also solve, but have solved in different ways, without, as far as is known, without, as far as is known, that's a big if, an even analogous structure, let alone a homologous one. In other words, this may be a uniquely vertebrate solution to a problem faced by all animals. If so, this would relate to Paul's introductory remarks Monday morning. One of the strategic points he emphasized is the importance of finding and understanding common design principles, the kind that evolution tends to conserve once it has hit upon them in its meanderings through the search space of life forms. The roof of the midbrain apparently houses an implementation of a common design principle of vertebrates, and vertebrates alone, conserved from Steingrilner's lampreys to ourselves. And so it is of interest to try to understand what that structure actually is for. What does it do? So let's take a closer look first at the structure itself and its connectivity. Here we're going to look into this little bump, and immediately at an intermediate scale we see that there's quite a bit of structure there. This is the magnificent structure that is housed in those two little bumps. First of all, on the afferent side, its afferent connectivity is organized with exquisite laminar organization. It is a cortex-like structure, but unlike cortex, it has, in a sense, judging by its afferents, it has many more layers than cortex. It has 7 cytoarchitectonic layers, from superficial down to deep colliculus. They are numbered, but into those layers come the afferents from different parts of the brain. 40 subcortical structures project to the colliculus, and in the cat, 25 cortical structures project to it. So 75 structures in the brain project to the structure in exquisitely laminar fashion. So here you see the different afferent sources. Here is the retina. Here are the primary visual areas, the early visual areas, layer after layer. Of course, if I showed all of them in their full extent, any one layer, this layer here, for example, from from area 18, extends, of course, across the whole colliculus. But I'm only showing a tiny sliver, a tiny sector, in order to indicate its depth in the colliculus. And then the other areas, 17, 18, 19, 28, 20B, 21A, 21B, and so on, down to higher and higher cortical areas, including frontal areas. And as you can see, they differ in depth. And here this lamination is exquisite. Here you have a number of cortical frontal eye fields. The lateral frontal eye fields are flanked by the medial frontal eye fields. What on earth does that mean functionally? We will see reasons for why this exquisite tessellate, this lamination is arranged in this way as we go along. Here you have the, where is this? Somewhere is the cingulate. Here is the cingulate. And here you have some of the frontal areas, and they extend to the very deep colliculus, and they don't care about the border between colliculus and periaqueductal gray matter. They extend right down into the periaqueductal gray matter, which may be considered, in a sense, as even deeper layers of the colliculus. And lateral to the colliculus have the midbrain reticular formation. This is the afferent side. 40 subcortical structures project to it, and 25 cortical areas project to it. Primary areas and higher order areas. Here is the efferent structure of the colliculus, equally amazingly arranged. The efferent structure is primarily from the intermediate layers of the colliculus. Here is the intermediate gray, and if you look at the intermediate gray in histochemical stains, you see a beautiful tessellated honeycomb pattern that the efferents form. The histochemistry, the efferents are compartmentalized within a common premotor structure, a common premotor map. There is a concatenation of different output channels, which are histochemically coded. These output channels are things like an output channel for catching a stimulus, moving the eye towards a stimulus. Another output channel is for escaping from a stimulus, a visual threat. So these are embedded together by this tessellated output structure in the same intermediate gray, but they end up in different targets in the brainstem for the output. And also upstream, they project to the forebrain. We'll learn more about the anatomy soon. So let's go through and look at some of the elements a little closer. Here are the cellular elements that compose the colliculus or tectum in different animals. Here's the frog. You see this cortex-like structure. They are even a little bit like pyramidal cells with a cell body with an apical dendrite. That becomes also in the sparrow. You have this pyramidal cell-like organization of the cells. In mammals, they are not so conspicuously pyramidal cell-like, but because this is rabbit here. But this one resembles a little bit of pyramidal cell. Here's intermediate gray and these star-shaped output neurons, which you can see here in cells filled after intracellular recording. They fill them with dyes and a map in detail the dendritic tree and the axonal trajectories. This is Moskovakis studies in the monkey. That's one contingent of output cells here. These are downstream projecting to the brainstem. These are upstream projecting to the forebrain via the thalamus. And Here are some of the interneurons with elaborate axonal branching trees that communicate between the different layers in the colliculus. Most of these are inhibitory, GABAergic inhibitory cells. Here's a little schematic diagram of the layers and the cells. Here are the inputs with their different depths, different input sources with their different depths, and here are the output neurons. This spectacular neuron, for example, is rather specific to the colliculus. Here is one at close up, and as you can see, the span of this dendritic tree, which sticks its final tufts up in the stratum zonale to pick up some information up in the stratum zonale, carried down to the cell body, pretty much like the big branching dendrites of the apical tufts of layer 5 pyramidal cells in cortex, but much, much bigger, much more widespread. It covers about a quarter of the entire mediolateral extent of the colliculus, this one tree. And now imagine these guys sitting in an array, overlapping next to each other. You are doing some integration here, and these cells explicitly project up to the pulvinar. So there is a lot of structure here, a lot of sophisticated anatomical structure. On the face of it, it seems rather implausible that a structure with this degree of structural complexity and refinement, both as regards its connectivity and its intrinsic organization, would do no more than to bring the eyes onto a target that has appeared in sensory space. A housefly routinely directs its body orientation very accurately to ambient targets with the help of a brain whose total size is no more than a speck of tissue weighing 0.4 of a milligram, composed of a total of 340,000 neurons. Considerably less than that number would be involved in the fly's orienting behavior, so why would we need 5 million collicular cells to do the same thing for our eye? Doesn't make sense. Yet this was the notion that dominated the conception of collicular function ever since Adamik in 1870 reported triggering eye movements by electrical stimulation of the superior colliculus. This became the visual grasp reflex or foveation conception of a collicular function, according to which the sensory and motor maps of the colliculus are matched in such a way that a stimulus-evoked local activation of the collicular sensory map drives an underlying local population of premotor units in the collicular motor map such that the line of sight or the fovea is moved onto the evoking stimulus. The eyes are straight ahead, here comes the stimulus, bing, the colliculus moves the eyes to the target, to catch the target and foveate it. In principle, such an arrangement might be implemented with a single generalized sensory motor mapping, as in this cartoon I made, inspired by Paul Churchland's simulated crab robot. You may have seen his crab robot toy. So here I've simulated it for the eyes. You send the information to the eyes to some kind of module which extracts one single location. You send that to a metrically deformed sensory map, and you project that sensory map onto a motor map which moves the eyes. Here you have the lateral rectus and the superior rectus, and you should be able, by this arrangement, a point-to-point mapping in two different matched sensory and motor spaces, move the eyes to target. So the many topographic maps superimposed one upon another in the colliculus go way beyond any such simple remapping. The colliculus features topographic maps of every sensory modality on which an animal relies for directional guidance. As a minimum, that includes vision, hearing, and somatosensory, touch. In addition, infrared, magnetic, electroceptive, and sonar senses in animals that use such exotic modalities. Whatever an animal relies upon to orient in space directionally in the moment is piped to the colliculus. And for the olfactory people, I would say olfaction, of course, is a sense whose directional uses for directional, for tracking, is very, very different from the appearance of a stimulus, a sound or a stimulus in the visual field or in auditory space. Olfactory tracking is a sampling thing, a sniffing where you're sniffing gradients. So that it does not project directly to the colliculus like all these other senses. But I bet you that in the hammerhead shark, which has this huge paddle that is growing out from its nose, and its olfactory, its nares, its olfactory receptors are sitting out here at the ends of this. they are presumably doing very, very quick directional detection of differences between left and right. And in the hammerhead shark, I would stick my neck out and say, perhaps you will find a more direct connection to the colliculus in the hammerhead shark than in any other living species. That's just a little bit of prediction here. If I'm wrong, I will be wrong. Since all these sensory maps are superimposed in laminar fashion within a shared premotor framework in the colliculus, it stands to reason that at least some form of sensor fusion must be taking place there. This in itself is not a trivial matter, given that eye and head movements bring the collicular sensory maps out of register with one another. For example, the ears are fixed to the head, while the eyes are not. The eyes are moving in their orbits. So a compensatory coordinate transformation is needed to keep the visual and the auditory space maps aligned when the eyes deviate from their primary position. So if I'm looking there, the relation between a stimulus, its retinal location, and an auditory stimulus will be displaced because the eyes have moved. This is indeed what takes place. David Sparks found in the colliculus that the spatial representation of a sound source in the colliculus varies with eye position. So there's a compensation for eye movement in the collicular representation of an auditory source. But sensor fusion cannot explain why the colliculus represents so many cortical areas, including some non-sensory association areas such as the cingulate cortex and also motivational afferents from sources like the hypothalamus. If all that's going on down there is sensor fusion, you don't need the hypothalamus to inform you. On the face of it, this makes it unlikely that any straightforward sensory motor account will exhaust the functional competence of the colliculus. So the question is, what does it do? It was in 1974 that Wirtz and Moeller, on initially rather scanty evidence, suggested that the colliculus somehow is involved in selecting the target for a saccade. Saccade is a rapid, quick flick of the eyes, a rapid eye movement. This suggestion opened the door to a new phase of colliculus studies concerned with how the colliculus might be engaged in the various operations that necessarily must precede the triggering of any given saccade, if that saccade is to be usefully directed. Selecting a target for a saccade sounds innocent enough, yet it may be regarded as the principal, primary, and final output of the entire cerebral machinery. This because in all instrumental behavior, the gaze, carrying foveal vision to locations of interest, as we look around the environment, precedes the rest of behavior like the acquisition marker of a laser spotter in a combat zone in modern warfare. Or if you're playing video games, you may have some laser spotter guy who marks the tank or marks the cave or marks something with a laser dot, and in comes the Air Force and blows the thing up. The gaze, the fovea, the line of sight precedes all other behavior exactly like that laser spotter. And if I can get this to work, it was shown in exquisite detail by Roland Johansson and colleagues working with Randall Flanagan. They performed fine-grained monitoring of the gaze as well as hands and fingers during manipulative activity involving small building blocks in the lab, and found that the hands and the fingers follow the flicking movements of the gaze as if hands and fingers were attached to the gaze by elastic bands. What you will see when we get this will be small. I wasn't able to download this. They have locked the download. So I will run it. What you will see here, this marker, this cross, this little white cross, is the gaze. The hand will come from down here. The hand will be very big. It will be picking up this block, touching up here, circumventing this barrier, and returning the block to where it started. The gaze all the time will precede the hand. So it will be like this: gaze, hand, gaze, hand, gaze, hand, gaze, hand, gaze, hand. That's how it works. The gaze precedes the manipulative activity, arm and hand and finger movements. So let's look at it. First, it will be fast, and you will have a hard time seeing it. Then there will be a slow motion replay with stylized gaze and hand. So let's look at it. You hardly saw the marker. Now they are replaying it. Gaze up here. The gaze prior to the movement, to the arm movement, hand movement, fixates every significant feature of the environment having to do with that movement. I'll play it again. Okay. Right. And I might as well load the next one because that takes a long time. There's other evidence as well that the coupling of arm or hand to the gaze is the brain's default mode of operation. And the so-called gain fields discovered by Anderson and Mountcastle in the parietal cortex during reaching behavior can easily be likened to the elastic bands in the analogy I just made. I said the fingers and the hand appear to be attached to the gaze as if by elastic bands. Well, the elastic bands are the gain fields that Anderson and Mountcastle discovered decades ago in the parietal cortex by analogy. We are now so while we are on the topic of the gaze movement, let me note that the colliculus is not essential for the act of moving the eyes. You should know that. For ocular motility itself. Pedro Pasic and colleagues in the 1960s showed that a monkey can move its eyes to a sensory target after its colliculus has been removed. So it is in no way essential for ocular motility as such. Even sensory guidance of the eyes is possible without it, as Peter Schiller and others later confirmed. It is, however, one of a number of loci that trigger eye movements. But typically such eye movements, if the animal is not artificially restrained, are only one part of the much larger behavioral constellation triggered by collicular activation. It includes every component of the behavior involved in the orienting reflex as classically defined by its pioneering student Sokolov and others. That includes eye movements, head movements, and trunk movements, like Sten Grillner pointed out. It includes pinna movements in animals that move their ears, and it includes the autonomic and the cerebral activation aspects of the orienting reflex as well. Cortex gets activated, flat EEG, and the autonomic GSR, skin conductance response, gets triggered by collicular stimulation. All the components of the orienting reflex are evoked by collicular stimulation. In fact, the superior colliculus is the lowest, most caudal point of the neuraxis, at which local stimulation will evoke a full and integrated orienting response. It would be tempting to call the colliculus let's see, this takes a long time to load, it's 22 megabytes. Somebody scanned my thesis into an MIT server. Very thankful to them. So it would be tempting to call the colliculus the central pattern generator of the orienting reflex in Sten Grillner's terms, were it not for the fact that it does not actually specify the particular moment-to-moment sequence in which eyes, ears, head, and trunk or limbs combine to produce a given orienting movement. The interplay among components of the orienting gaze shifts is apparently settled downstream of the colliculus, as Sparks has argued. There are elaborate brainstem connectivity bundled along the medial longitudinal fasciculus, which carries the vestibular, the cerebellar, and the postural information, including eye position information, integral to the fluid interplay of the various motor systems involved in orchestrating an orienting response. Even after having expanded the collicular role to that of triggering the full-blown orienting reflex, which is the principal mode of spontaneous foveal acquisition of targets of interest in all higher animals, we are not done. Without the colliculus, animals do not exhibit escape reactions to visual threat. Such an escape behavior reorients the animal away from the eliciting stimulus. So a visual grasp reflex conception of collicular function is useless for the escape to explain the escape behavior that manifestly is implemented in the colliculus and can be triggered from it. no orienting toward that stimulus need precede the precipitous escape triggered by effective visual threat. As I had occasion to document by frame analysis of hamster behavior, they were filmed while the animals launched explosive escape responses to overhead visual threats in a specially designed testing arena. And it managed to load it before I was done even. So here is my testing arena. I built at MIT a huge Disneyland for hamsters. Might as well show this. I'm so proud of it. They lived permanently in my testing arena. This is the back of the testing arena. I had them in a hamster hotel in 5 compartments. There were 5 hamsters living here permanently, and each one had a light cycle offset by 1 hour. So I was programmed totally, like Leah Krubitzer said, I was programmed to run these hamsters. Once an hour, a new hamster entered the arena. Its light cycle was now, they were all on the same light cycle, staggered by 1 hour, and I had to sit there and run the experiments. The hamsters basically ran the experiment themselves by triggering various automated stimuli while they walked through this arena. Here is the arena seen from the top, and I will show the whole thing like this. So here is the arena. The hamster comes in here. The hamster opens a door which turns on the overhead camera, which films them. This was on Super 8 film way back. It was not a video. It was reel to reel. The hamster triggers, and while it walks here, various light beams are being broken by the hamster, or it releases various stimuli. It goes through a maze here, and finally comes out to an open field. And very rarely, once every few weeks, because if you show a visual threat without consequence to a hamster many times, it will not work. If you show it only once, rarely, once every few weeks, a silent visual threat comes in, a black board swept in over the cage, over the open field. The hamster is heading for a sunflower seed sitting here. And while it is progressing towards the sunflower seed, the black board sweeps in silently overhead. At that point, the hamster explodes. It doesn't arrest. It immediately, from progressing forward, it breaks into a scrambling escape which is so dramatic that sometimes they tumble over themselves and roll. They are throwing themselves like this and scrambling down 3 escape routes, one on the right, one on the left, and one where they just came from. They want out from there because this is, a raptor. This is a bird trying to catch them. So now the next one is my frame by frame. Here they are, frame by frame, tracing of their escape. So here is the exit into the open field. There is the little marker where the sunflower seed sits. Here they came out and entered the open field, and this is the edge of the threat overhead. Here are the initial head positions of the hamster marked from the frames of the film, and this is the escape trajectory. To see exactly what is going on, here I have made the resolution twice. This is 10 centimeters. Here's I have taken all the head positions, collapsed into one head position, and see where they head. First marker is at the 168 milliseconds after stimulus appearance. Some of them, even within 168 milliseconds, have initiated their escape. Others require another 168. Within another 168 milliseconds, they are gone. They are bolting back here, and there is not a trace of an orienting movement towards the stimulus. They don't even arrest. And it would be stupid for a hamster, wouldn't it? A threat comes, and it goes, looks up at the threat. No. That's enough milliseconds to get killed, okay? So they have to bolt. The colliculus does this. When I undercut the colliculus, absolutely no reaction to the overhead threat. I undercut the colliculus, severing its efferent connections. No response to visual threat. They simply went on to pick up the sunflower seed as if nothing had happened. 1 or 2 animals hesitated a little bit and then proceeded to pick up the... It's an astounding difference. So it's definitely based on the colliculus. Okay. Now, there we have it. Off with that. Okay. But the colliculus is not the only brain locus engaged in gaze and orienting movements. At least half a dozen cortical areas are functionally specialized for the control of gaze movements. Of these, the principal ones are the frontal eye fields inside the arcuate sulcus of the frontal lobe and the parietal gaze area in the lateral bank of the intraparietal sulcus of primates. And I will call these two principal areas the cortical gaze fields for short. Since the colliculus is not alone in the game of gaze control, if we are to understand its specific role, it must be placed in this wider context of the brain's gaze-related circuitry. So first, a highly schematic diagram of the major players in that game. This is from Douglas Munoz's group. He has placed the colliculus quite appropriately, in my view, right in the middle. Here you have the dorsolateral prefrontal cortex, frontal eye fields, lateral interparietal area. These two together I call the cortical gaze areas. V1, you have pulvinar, mediodorsal nucleus, actually it's parallel portions, and the basal ganglia, all interacting with the superior colliculus, and then output to reticulospinal system for eye and head and even trunk movements. Because when a sound goes off behind you, the head is not enough. You have to do this. You have to move your trunk. And it's exactly the same in humans as in lampreys. When Stein-Grillner stimulates the caudal-most colliculus in lampreys, their whole body turns. Same with us. If you get the caudal-most edge of the colliculus, our whole body will turn. So it was while I was trying to relate the colliculus to the circuitry by which the cortical gaze fields operate that I finally was shown the picture on the puzzle box of collicular function. I had a little brainstorm. That's why I'm telling you this whole long story. The crucial information was contained in the final two sections of a 1986 paper by Huerta, Krubitzer, and Kaz on the subcortical projections of the frontal eye fields, rigorously defined by cortical microstimulation. Here is the paper. I printed it out, and I had hoped that Leah would be sitting in the audience because this is one of her early papers. When she was working with Kaz, that's where she started her career, and we heard her the other day, and I'm waving her paper from 1986 because I read it a few months ago, and it suddenly clarified the whole picture. In the final two sections of this paper, Herta, I asked Leah who wrote those final two sections. Herta starts expanding and saying, well, let's not look only at the frontal eye fields, but the parietal eye fields and how they are projecting downstream to the subcortical structures. And this whole paper is about the subcortical projections from frontal eye fields. Herta pointed out that the sizable set of subcortical nuclei that are picked out by receiving projections from the cortical gaze fields is rather consistent across higher primates. And indeed, in outline, it holds across mammals generally. These subcortical targets of the cortical gaze fields tend not only to be connected to one another, but to the superior colliculus, which also receives direct cortical gaze field projections. The colliculus then, in turn, projects to these cortical gaze field targets and via a synapse in the thalamus to the cortical gaze fields themselves. So this is highly interconnected. This places the superior colliculus in a peculiar duplicative and parallel position with respect to the gaze field-related circuitry, a position I have summarized in a diagram in my recently published paper, as follows. There is the reference to the paper I just waved, Herta, Krubitzer, and Kaz, 86, Journal Comparative Neurology. And here's my diagram, put together essentially by inspiration from that paper. And it's complex, but with the help of SketchUp, that free Google perspective drawing program, I managed to produce this figure. This is all very schematic. This is the cerebral cortex. And I've organized this in tiers. Here is tier 1 of subcortical projection targets from the cortical gaze fields. Here is tier 2, subcortical targets of the gaze field projections. Tier 1 contains dorsal thalamic and basal ganglia targets from the cortical gaze fields. Tier 2 contains ventral thalamic and midbrain targets of the cortical gaze fields. When you arrange it in this way, you see a pattern in that the cortical gaze fields are distributing their efferents to both cortical gaze fields. The frontal gaze field and the parietal gaze field are picking out basically the same structures. The paralamellar portion of the mediodorsal nucleus, the pulvinar, and a complex of nuclei called the extended intralaminar complex, which at its caudal end includes nucleus limitans and suprageniculate nucleus, then the classical intralaminar nuclei, and the paralamellar portions of the mediodorsal nucleus and the VAVL complex. So this is called the extended intralaminar complex. These are apparently all oculomotor related, and not only oculomotor but cephalomotor, the whole orienting reflex. When we go to naturally the basal ganglia. So I have marked all the direct projections from the cortical gaze fields by curved lines. So you see they project directly to the colliculus. They project directly to the brainstem oculomotor circuitry. Actually, it's reticular premotor circuitry. They project directly, these are also curved lines, to these mediodorsal, pulvinar, VAVL, extended intralaminar complex, and on. And I have distributed by buses, not to clutter the picture, subcortically in the ventral thalamus and midbrain zona incerta, rostral intermediate nucleus of the medial longitudinal fasciculus, midbrain reticulofascian, ventrolateral geniculate, and pretectum. If you look closely at this figure, you will realize that there is hardly a way to get out of tier 1 except through the basal ganglia. All of these nuclei that receive frontal gaze field and parietal gaze field projections in turn send their output either back to cortex, which we are not interested in. We want to get down to the output, to the motor parts of this. They send it either back to cortex or they send it to the basal ganglia. Here they gather 1, 2, 3, they're all sending them to the basal ganglia. How do you get out of the basal ganglia? Most of the people are interested in the loops that go back to cortex. We're not interested in that. We want to get out to the motor system. From the basal ganglia, they go directly on the primary output pathway of the basal ganglia, which is to the substantia nigra, pars reticulata, and lateralis. Basal ganglia to substantia nigra. How do you get out from the substantia nigra? You don't go downstream, you go straight up to the colliculus. And from the colliculus, you go downstream. Same thing with pretectum, ventrolateral geniculate, MRF or IMLF, and zona incerta. They receive projections from the frontal gaze fields, and they project to the colliculus, and that way out. They also, these differ from tier 1 ones, these have their own independent downstream projections. So you can get, what you should notice from this figure is there are multiple ways in this system to get to the brainstem circuitry, which I have marked as MLF here, medial longitudinal fasciculus. That's a little exaggeration. It is a bunch of fiber tracts that are bundled around the medial longitudinal fasciculus. That's the earliest fiber tract to develop in the brain. It's the mainstay of the whole brain. Everything in embryology, it's fiber tracts start with the MLF, and then all the fibers accumulate in various ways of branching off from there and so on. The first to be in place in the brain is the MLF, and it's central in the sense that it carries all the vestibular, visual interactions. It carries the basic orienting, postural orienting circuitry of the brainstem, medial longitudinal fasciculus. So you can get to the medial longitudinal fasciculus directly from the frontal gaze fields, here, down to the brainstem, via colliculus down, via not here, but here, basal ganglia, substantia nigra, colliculus brainstem, or frontal gaze fields to rostral intermedial MLF and downstream, or to the frontal gaze fields, parietal, let's say, to the MRF. Here comes a connection, MRF, and from there downstream. So there are multiple ways to get downstream. Not all of them go through the colliculus, but for any place that you look here, any place you try to get downstream, You are never more than 1 or 2 synapses away from reaching the colliculus and being reached by the colliculus. And now people who are into graph theory should start smelling a rat. Here you have, smelling a hamster maybe. Here you have something that looks like a graph theoretic hub. Well, it's even a little better than that. In informal terms, I have not done the quantitative stuff. In informal terms, the colliculus looks like a super hub of the gaze field related circuitry of the entire brain. What we see here then is that the colliculus forms a connective hub arrayed both in series and in parallel with the connectivity between the cortical gaze fields and the brainstem circuitry for control of orienting. This curious duplicative parallelism, noted first by Herda and colleagues in this paper, this is what they pointed out in those last 2 sections, that everything goes to and comes from the colliculus, but it's not an obligatory path, but it's always an optional path. It means that from virtually any component of the supranuclear orienting circuitry, one is but 1 or 2 synapses, as I said, removed from the colliculus, from which any other component of that circuitry can be reached on equally short paths. The colliculus is positioned like a spider in its web amidst the multiply connected subcortical circuitry for control of orienting. And I have placed it like a spider here. Sitting here, I've just elevated it and shown all the connections that radiate. Most of these are bidirectional. A few of them are monodirectional. This is bidirectional, mutual, afferent and efferent, multiply connected. And to get back to the gaze fields themselves, the cortical gaze field from the colliculus, you need a synapse in either MD or pulvinar. But it's only 1 synapse and it's direct. It's 1 synapse in between in the pulvinar. Whatever path one chooses to descend along, one is never synaptically far from reaching the colliculus or being reached by it. In informal graph theoretic terms, the colliculus appears to form a connective superhub of the subcortical orienting circuitry. As such, it may perform a more indirect or higher order function than its midbrain location might suggest. When you see a superhub, you know that you can do things that are hard to do in other ways. A function which otherwise diverse components of that circuitry have reasons to access and be accessed by. Everybody in the gaze control circuitry seems to be interested in both talking to and being talked to by the colliculus. So what might that function be? Why are they all so interested in the colliculus? The wide-ranging afferent and efferent connectivity of the superior colliculus indicates that it must perform an integrative function of wide scope. Here we have just the cortical afferents, 25 afferents, add 40 subcortical ones. They're all ending up in one place in a laminar array. Something is going on here of a highly integrated kind. Remember this diagram leaves out some 40, like I said. You have to add 40 layers to this. A key to its integrative function comes within reach by asking what it is that its 2 principal behavioral outputs, namely spontaneous orienting movements towards a target and visually triggered escape away from a threatening stimulus. What might these 2 have in common? Functionally, there is very little common ground between orienting target acquisition and escape from visual threat, except this. In both situations, the brain selects a spatial target of immediate behavioral priority. Now, immediately, do it, either away from or towards. In the case of escape from visual threat, that target isn't even in the visual field. It's a remembered location from the familiar arena which the hamster has traversed for hundreds and thousands of trials. They know where the escape routes are. They know where to get back to the nests or get into the shelter of the maze that is built in the middle of the arena. So it's a remembered target they are heading for. But they must pick that target. They must remember it right at the split second in which they escape. A so far elusive generic definition of collicular function may accordingly come within reach by focusing on the determination of target priority. rather than on either the eliciting stimulus or the nature of the resulting movement. Priority in this connection is to be distinguished from salience. There's been a lot of talk about salience in the colliculus, but salience is only bottom-up. Priority is top-down interacting with bottom-up. Priority involves contextual, motivational, and goal-related factors in an interaction between top-down and bottom-up influences. This is exactly the kind of functional construct that might make sense of the conspicuous convergence of cortical information with primary sensory afferents in the colliculus. Top-down and bottom-up. Who here knows about the Lovejoy and Krauseless experiment on the colliculus? This is just to illustrate the point. If you had asked me more than 2 years ago, how does the brain do top-down biasing of a visual search task? You're confronted with an array of stimuli and you have to pick the red squares. And there are red triangles and there are green squares and there are all kinds of colors and shapes. You have to pick the red squares because you like Moscow. That's your task. You have gotten instructions from the experimenter, pick the red squares, so you have top-down biasing of attention. And with the instruction, the top-down biasing makes you better at picking out the red squares. Your reaction time decreases. So, if you had asked me a couple of years ago, how does the brain do that? The answer is obvious. We know about the feedback projections from frontal cortex going all the way back to V1, a whole cascade of feedback projections. Everybody has talked about those feedback projections as the primary route of sending top-down biases onto the sensory system to do exactly this kind of attentional biasing, top-down biasing. Krauselitz set up trained monkeys in a visual search task of exactly this kind. They were trained, so they knew the instructions. Pick the red squares, let's say. Well, those weren't the stimuli, but exactly analogous to the human visual search task. He had an injection cannula in the colliculus. The monkeys were trained to criteria 90% or something, whatever his criterion was, and then he injected a little muskimol into the colliculus, basically deadening a local area in the colliculus, and the task completely breaks down. Top-down biasing of sensory attention does not go by the obvious route, namely the frontal eye field and dorsolateral prefrontal cortex feedback connections back to the sensory areas. It goes from the frontal cortex down to the colliculus and back up to the sensory systems via the pulvinar, because it's wiped out by a small collicular injection of muskimol. That surprised me. That surprised me no end, because I would never have guessed it, even though I'm very partial to the colliculus. A range of empirical evidence for a priority map interpretation of collicular function has been accumulating over the past decade in single-unit studies of the colliculus of behaving macaques by Douglas Munoz and colleagues, as well as Robert McPeak, Jeffrey Schall, Richard Krauselitz, who I've just mentioned, all working on the monkey colliculus, and Eric Knudsen working on the barn owl optic tectum. Same thing holds for the barn owl optic tectum. It's priority, not salience. Munoz, for example, has shown that the magnitude of the transient visual response that precedes the motor-related burst of intermediate layer cells has the right properties to carry a priority signal. When large enough to outcompete other collicular bids for alternative priority locations, it triggers an orienting movement whose reaction time is proportional to transient response magnitude. It has other essential attributes expected of a priority signal, such as context and task sensitivity, dependent on top-down biasing, and it habituates on repetition when not followed by additional consequences. At magnitudes below that required for triggering an orienting movement, the transient response is associated with covert orienting of attention. And it's well established by now that the colliculus is an essential structure for the covert direction of attention. That which we do while we keep our eyes fixed, we don't look at the thing, but we attend over there. We don't want anybody to notice that I'm looking over there. I can put my attention there, covert attention. The colliculus down in the midbrain is crucially involved in covert attention. Of course, many other forebrain gaze-related areas exhibit priority-related neural signatures, but the picture that is emerging from the labs I named is that the final Correlation and competition between these various sources takes place in the superior colliculus itself, as those sources place their bids alongside those of their forebrain colleagues via the direct projections to the collicular intermediate and deep layers. Early visual areas converge on the superficial visual layers of the colliculus, where it is areas higher in the cortical hierarchy, notably the parietal gaze field, the infratemporal cortex, the frontal gaze field, and the dorsolateral prefrontal cortex that project to the multimodal and premotor intermediate layers of the colliculus, where these authors are finding the priority signal. And that is, it's close to the collicular output. This is where it has to get. Because that all of this I have been talking about is very short range. It is within 100 few milliseconds. Our gaze moves 3 times per second. 5 times per second when we read, but normal viewing 3 times per second. So you have a couple of 100 milliseconds to decide where is the next eye movement going to be. You don't want to waste that next eye movement. You want to do it just like in the Flanagan movie you saw. It's got to be placed exactly. So to get that right, you have to collate all the relevant sources. And the amazing thing is they are collated and compete with one another down in the midbrain. And they do it by this elaborate connectivity, which sends all the relevant sources of information, the highest levels of the brain, dorsolateral prefrontal cortex and so on, down to the lowest primary sensory afferents, converges here. 40 subcortical defense. The retina projects, of course, directly to the colliculus. What this work suggests to me is that the anatomical structure and connectivity of the superior colliculus provides a comprehensive mutual interface for brain systems carrying information relevant to defining the location of high-priority targets for immediate reorienting of receptor surfaces. At that interface, that is in the colliculus, through interactions within and across its lamina, these systems settle their several bids for such a priority location by mutual competition and synergy. The result of this competitive process is a single momentarily prevailing priority location subject to immediate implementation by deflecting behavioral or attentional orientation to that location. The key collicular function, according to this conception, is the selection on a background of current state and motive variables of a single target location for orienting in the face of concurrent alternative bids. In this capacity, the colliculus would serve as the brain's final priority comparator or priority gate for immediate reorienting. That's extremely short term. You have 300 milliseconds to reorient. It would determine which of simultaneous bids for an orienting movement should prevail in gaining momentary control of collicular output circuitry housed in its intermediate layers. That includes, when nothing competes, continuing the current orientation unchanged, which helps explain what the fixation neurons in the anterior colliculus are all about. They're about maintaining fixation. Don't change. And they are sitting there clamping the gaze to its fixation spot. So let me round up. How much have I taken? How much time have I taken?
You've got a reasonable amount of time.
I'm rounding up. No cortical...
68 minutes.
Okay, yeah. So this is, I'm heading to the end. No cortical gaze field is as directly connected. Now the question is, why isn't the frontal eye fields or the parietal gaze field doing this? Because no cortical gaze field is as directly connected to as wide a range of sources carrying information bearing on the decision where to turn next as is the midbrain superior colliculus. The cortical gaze fields receive high-level information, but not primary sensory afferents, while the colliculus receives both the latter and the direct output of the cortical gaze fields and numerous additional cortical and brainstem afferents as well. Its broader afferents enables its intrinsic circuitry to weigh a wider range of information bearing on the very next orienting movement than any other known neural system, with the possible exception of my other favorite in the brain, the zona incerta, which sits next to the colliculus, just a little bit above it, in front of it, with which the two are reciprocally connected. just like the colliculus is reciprocally connected with the parabigeminal nucleus. This predicts that without the colliculus, an animal will be capable of turning and orienting, but not with as comprehensive a moment-to-moment weighting and comparison gating of all relevant sources of information as when in possession of an intact colliculus. So this explains the indistractability of colliculectomized animals. The advantage of conducting structured interactions between low-level primary afferents and high-level cortical information in a compact convergent laminar mechanism is twofold. First, this way the brain escapes the liability of entrusting moment-to-moment decisions to an executive fed only highly derivative information, like most of our politicians, at least when they are in executive positions. It escapes the liability of entrusting moment-to-moment decisions to an executive fed only highly derivative information. When high-level cortical areas place their priority bids with an independent priority comparator, the colliculus, the brain as a whole, through the collicular offices, stays open to last split-second course corrections, even by low-level sensory information, provided its magnitude is sufficient to override current competitors. It is worth noting in this connection that cognitively demanding high-level deliberations are often readily postponable, compared with intrusive sensory change that might spell disaster unless immediately attended to. Though often a fleeting glance is all that is required before ongoing behavior can be safely resumed, without that fleeting glance you might be dead. Though often a fleeting, these cautionary glances nevertheless compete with the demands of ongoing behavior for task execution. Both utilize the same effector equipment for orienting, hence the need for a mechanism to resolve conflicts between them. So, first advantage of putting the comparator in the colliculus is you stay open to low-level course corrections in case they should turn out to be crucial. Most of the time they are not. That's why a monkey without a colliculus looks totally indistractable. With a colliculus, it's glancing all about all the time. It's doing those cautionary glances. And it looks like a vacant stare without a colliculus. The second advantage of putting it down there instead of letting the frontal eye fields or parietal gaze fields do it is by sending all priority bids to a compact neural space where they compete by means of short axon intrinsic connectivity. In the colliculus, it's a compact, small structure, those two little bumps. It interacts through short axon intrinsic connectivity. The interactions needed to determine target location can occur far faster than anything that might be accomplished by long-range cortical-cortical interactions among multiple systems. Because it's got to be among multiple systems. There are multiple bids in the forebrain for where to look, because the cortical areas are specialized. One might say, oh, this drifting stimulus is the one. I love that one. I want you to look at that one. Another area says something different. So because multiple systems have to interact, long-range cortical connectivity back and forth among those systems is a huge waste of time. You want to do it in a compact space where you can do it super fast by short axon connections. The abolition of short latency gaze shifts by lesions of the colliculus or its local inactivation accordingly may reflect the absence of the rapid decision-making competence by which the colliculus normally drives the orienting machinery, rather than, as it sometimes has been interpreted, a mere quantitative slowing of the orienting system, which doesn't make sense to me. So now you can understand, against this background of the functional interpretation of a central compact comparator space where all the bids for the next priority or where to look are being compared and settled for immediate execution downstream. So now we can understand the lavish connectivity both of the afferent and the efferent sides on the colliculus. Given such a function, it stands to reason that it must receive afferents from every neural source that actually has something to say about where to look and attend next. So with that, I hope I have shown you at least one possible way of making sense of the exquisite circuitry and connectivity of the laminar and tessellated structure that is housed in the two bumps oops, there it is, 2 bumps that crown the midbrain, and which Thurston Lacali called the quintessential identifying feature of the vertebrate brain. If the interpretation I have given you should turn out to be on the right track, the lowly midbrain location with its orienting superhub, a location which in a sense minimizes its distance from all parts of the neuroaxis. Everybody can reach it, and it can reach everybody. High or low may help account for the extent to which vertebrate decision-making is centralized compared to many other animal forms, and for the degree to which the vertebrate brain plan proved compatible with such a wild diversity of animal forms and adaptations in its long and fertile evolutionary history. I don't think there's another phylum with as much diversity within it. In the oceans, on land, and in the air, there are insects, there are some aquatic insects, there are terrestrial insects, and there are flying insects, of course. But in terms of the diversity of forms and the spectacular range of adaptations shown by vertebrates, they all have this little hub, this orienting hub, to rely upon as a comparator as they expand different parts of their machinery. There it sits, ticking away, making the optimal decision for where to look next, weighing all the evidence. Thank you very much for your attention.
Questions?
Yeah. Yeah, I have a question. If I understand correctly, you say that the superior curriculum is all about awareness, and the cortical region are about meaning, no? And so when we have a reflex response, which means, for instance, the predator that you were saying the animal was seeing,
yeah,
and the animal is able to connect, notice the predator and head toward the exit. It's well. As I was thinking myself, and I say, well, in order to understand the predator, I need to have a memory of the predator or something, and in order to know that where to go, I need to understand that in the exit I'm safe. No? And how does the curriculum questionate this in solo time without accessing to the superior function?
It's being fed continually by a single synapse from the 25 cortical areas, like in a cat. In a hamster it's fewer, but in a cat, 25 cortical areas. A single synapse. The colliculus is not more distant from the cortex than the cortical layers are distant from one another. You could conceive of the colliculus, because it's a monosynaptic direct projection from layer 5 of cortex, layer 5 pyramidal cells. You could conceive of the colliculus as a seventh layer of cortex, metaphorically speaking. So it's sitting right next to it. It's getting bombarded all the time. Now, the predator thing is not learned. The first time, these hamsters were bred in the laboratory. They had never seen a bird. They had never. We have nice animal keepers. They didn't scare the poor animals. So the animals were cared for, perfectly tame, used to human beings, used to being handled. One of them bit me, but. So when I kept them in isolation, in an open arena for weeks, there was a paper published the year before I started my thesis work saying, are hamsters visual animals? Because they had put hamsters on a little pedestal, on a little platform, like this. The hamster is sitting on a platform. They wanted to make a clean experiment. No extraneous stuff. Put it cleanly on a little pedestal, a little platform. Hamster is sitting there, and then they went like this, with discs, big black things, high contrast things, and tried to scare the hamster. And they were just sitting there, looking at them. Okay? There was no place to escape. Furthermore, the escape response, the basic escape response is built into the collicular circuitry from the retina, and it's maximized along the midline, which means upper visual field. There's a specialization in rodents. I don't think in all animals, but in rodents, there's a specialization. There was a Nature paper recently showing that, specialization for overhead detection of threat. It's innate, it's there, built into the system. So here are these hamsters who has never seen a bird or anything, never been threatened by anything, isolated for 2 weeks in an open arena with an escape route, a hole in the floor, and down there was the nest for several weeks. And I took a regular dustpan, a black dustpan, and waited till I could look over the edge, waited till the hamster was perfectly relaxed, eating sunflower seeds, and I took the black thing, moved it overhead, an explosion right down the hole. Okay, it got to remember where the hole is. They make an absolute straight beeline. It's so they know exactly where it is. How that information is sitting in the hippocampus. This is your part of your question. That information is sitting in the hippocampus. The place map in the hippocampus, where they get used to, the hole cannot be innately recognized. They must have learned where the nest is, where the exit is. Hippocampus and parahippocampal gyrus projects to the parietal gaze field, from there straight down to the colliculus. So that information is available via the parietal gaze field to the colliculus. But that information is sitting there all the time. It doesn't. The biasing signal on the colliculus is in a sense always available, and of course it must be retrieved. There is some milliseconds go. It took the fastest ones escaped within 168 milliseconds, which is pretty fast. But they are small animals; they tend to be fast. But anyway, some took a little longer, a couple of 200 milliseconds. So there's time to get from the hippocampus to parietal and down to the colliculus.
So I have a question. So the colliculus, I completely agree with your interpretation that it's the hub for multisensory integration in order to drive orienting movements and, like you say, escape movements, and quite a lot of other species-specific innate behaviors that may be coordinated there, like, for instance, face recognition in infant humans. Yeah. But the question is, when you have competing peaks in the collicular map, how do you resolve that competition? Yeah. And you pointed to intrinsic mechanisms within colliculus, but the obvious other candidate for resolving competition would be the projection by the basal ganglia substantia nigra back to colliculus. So there's a balance, I think, between the selection that might be happening within the colliculus between peaks and the selection that might be happening through what is clearly, we know from Sten's talk, an architecture that's optimized for selection via the basal ganglia.
I agree perfectly. I think that's the reason for why tier 1, you can't get out of tier 1, which is really the primary cortical projection down to the dorsal thalamus, pulvinar, MD, VAVL, and intralaminar. You can't get out of there. The primary route from cortex is to directly to the basal ganglia, indirectly via thalamus. Sten emphasized very, very well the fact that this thalamic route to the basal ganglia is massive. It's a huge one. And it's multiple nuclei are projecting, and they are all aligned sort of paramedian from the back to the front. They are all projecting into the basal ganglia, and from there. So here is a huge pre-selection. I think that the basal ganglia are settling a lot of the cortical decisions. The multiple bids from cortex are already, and now this straight axis up to the colliculus is simply the final, is essentially commanding the colliculus in those cases. If right when that is happening, some explosion comes in from the retina or something, there might be some ways in which you could deflect that, but all of them are placing their bids, and I think these inhibitory direct bids are among the strongest ones. In other words, these are priority channels, and the colliculus is incredibly clamped by inhibition. It is the most inhibited. It's sitting on a razor edge, essentially, perched on this incredibly massive inhibition from multiple sources. Let's see if I can remember them. The first one is the substantia nigra pars reticulata, massive GABA inhibition. Second one is zona incerta, massive GABA inhibition going right into the colliculus topographically. The third one is an area called the periparabigeminal area. The periparabigeminal area projects GABA inhibition to the colliculus, and there is a pedunculopontine zone Z or something, or X, or whatever that zone is called, a special zone next to the pedunculopontine nucleus, projects GABA inhibition to the colliculus, and the parabigeminal nucleus itself projects acetylcholine. Now this is the superficial colliculus, but anyway, acetylcholine via collicular intrinsic interneurons, which are inhibitory. So this, most of the interneurons identified turn out to be inhibitory. So this is an inhibitorily incredibly clamped structure. It's just sitting, suppressed, and waiting. So these multiple hills in the colliculus, there is room in the colliculus to have several things growing. You can have a couple of hills budding, and very soon one of them will be squelching another one. But I agree perfectly. And it's even nicer than that, because I very much liked what Stein Grillner said about the dual organization of the matrix and striosome thing, one for avoidance and one for towards. That goes right through the I think it goes through the basal ganglia circuitry, one part of the habenula thing for the avoidance, and that I think it's a general organizing principle in animal orientation, is that there's a segregation of towards and away from. And it holds for the basal ganglia, it holds for the colliculus. Charlie Gross has a paper on an infratemporal projection to frontal, which he suggests is an avoidance pathway. So it's a very nice theme. Yeah, I agree.
And another comment is exactly how intelligent is the colliculus, because in these multisensory integration situations, I believe Barry Stein's group have shown that you get, for instance, if you've got a movement over here and a sound over here, then you get a superadditive response in the colliculus relative to if there's just the movement on its own or the sound on its own. You get a response which is much stronger in the colliculus when they're both there than if you just sum the individual responses. And I believe he shows that's dependent on the cortical input. So that suggests that the colliculus maybe isn't that bright in terms of really prioritizing the information, that the convergence is there, but in terms of making smart decisions, certainly in mammals, the cortex is really helping you do that.
Absolutely. It's only the final step.
Yeah.
It's only the final step. You're right. They showed you lesion 18, I think, and it disappears. They did developmental studies. That integration is definitely cortical. The colliculus doesn't do it itself. But once the bit is placed there, this is the final sliver of time before you got to settle on where the eyes go. There, the colliculus opens the chance of aborting that good agreement between those if something else comes up. Yeah. That's how I conceive it. In other words, the colliculus is not only perched on the knife edge of inhibition, it's perched on the very edge of now. It's just its time horizons are a couple of 100 milliseconds. That's all.
Any other questions? Okay. Well, I think we've had a very intense morning, and we're all ready for a bit of a break and some lunch. Lunch is at 1:30?
Yeah, 1:30.
Yeah. Okay. So
with the students now, we will discuss the projects. So we stay for.
Okay. So students stay for some discussion of projects, and we all meet again in the usual place for lunch.
Yeah, lunchtime is there.
So let's say thanks again to this morning's speaker. Thank you very much.